that we have in one of the line of research we have in the lab right now, which is about understanding the role of CAR2 cells in the postnatal hippocampus. So one of the things that has always fascinated me is how the nervous system gets together. How do neurons know where and when to migrate? How do they know what type of neurons to become? And how do they know who they should talk to? So practically, how do we go from something so relatively simple at early stages of prenatal development to something so complicated in the adult where we have millions of cells and billions of connections? And if we look at a little bit at everything that is going on during development, we can see that there's very different processes happening. We have migration of neurons, neurons are differentiating, they are integrated in the circuit, and we have many different types of cells, right? We have glutamatergic neurons, different types, GABAergic, many different types of GABAergic cells. And then we have also this transient population of neurons that are only present during specific type of development. And they tend to be located in superficial layers or deep layers of the cortical region, such as, for example, the subplane neurons. And one of these transient population neurons that we have in cortical regions are the Cajal-Retzius cells. Cajal-Retzius cells are very well known for their role in prenatal development because at this stage they are the main source of reeling, and reeling is very important for the correct migration of pyramidal cells from the ventricular zone into the cortical plate and the formation of this really beautiful inside-out patterning in cortical regions. However, very little is known about these cells in postnatal development. And this is also probably due to the fact that most of the studies that look at circuit development are actually done in neocortex. And in the neocortex, Cajal-Retzius cells die soon after birth. Here is a marker, is a staining for P73, which is a marker of Cajal-Retzius cells. And I hope you can appreciate that by P15, the number of cells is drastically reduced, and by P30, they are pretty much gone. But this is not the case at all in the hippocampus, where the density of Cajal-Retzius cell is still quite high for several months after birth. If we just use a reporter mouse crossed with a transgenic line that I will introduce better in a couple of slides to label the Cajal-Retzius cells, we can see that they are localized in the hippocampus in stratum lacunoso moleculare of the CA region and in the molecular layer of the dentate gyrus. And I hope you can appreciate how the dendrites and the axons of these cells are very localized in these specific layers. They don't really cross that much the boundaries, although we do know that the axon can stand for up to subiculum and even the entorhinal cortex, right? And we do know that the maturation of the hippocampal region is prolonged during postnatal development. The hippocampal function takes a few weeks to emerge in rodents. So the Cajal-Retzius cells are really localized in the perfect place and at the perfect time to be able to contribute to the maturation of the entorhinal hippocampal circuit. If we in fact look at where the inputs of the entorhinal cortex arrive to the hippocampus, we have layer 3 cells projecting to the SLM region, layers of the CA regions, and layer 2 projecting to the molecular layer of the dentate gyrus. So understanding what Cajal-Retzius cells are doing in postnatal development has been something that has driven me for quite a few years at this point. But the first thing that you have to do when you want to understand and study one specific cell type is try to find a way of manipulating it or tagging it. So what we did is that we use the PD-1 secret line, which was created by at the Rockefeller University through the GenSat project. And we cross it with the TD tomato as a first thing just to characterize the line and using P73 and reeling, which has 2 known markers of Cajal-Retzius cells, and really see how good the line was for us in the hippocampus. And we saw pretty much 99.9% overlap between the 3 markers, really showing that this line is very, very good, specific and selective for Cajal-Retzius cells in the hippocampus. So at this point, we have the transgenic mouse line. What do we do with it? So what we decided to do is we decided to use this line to kill the Cajal-Retzius cell at early stages of postnatal development. So we take P0 pups of the PD-1 secret line, and we inject the virus in the dorsal hippocampus bilaterally. And the virus that we inject is the one you have here at the bottom. And this virus can drive the expression of two proteins, actually. M-cherry is pan-neuronally, so will be expressed in every single cell that is infected by the virus. But then the diphtheria toxin fragment A, DTA, is under the control of the lox site, and therefore only the CRIP-positive cells will undergo a recombination, start to express diphtheria toxin fragment A, and die in the matter of a couple of days after the expression of the DTA. Here's a quantification that we have done of how good the virus is, and it works really, really well. Here on the left, you have the CRI-negative animals, and on the right, you have the CRI-positive animals. We did the analysis at different developmental stages, but at the bottom, you can see the P73 staining. So again, the marker of Cajal-Retzius cells, and you can immediately see that the number of Cajal-Retzius cells is drastically reduced. We have around 75% reduction on average, which is conserved between the P15 and the P30 ages. So also suggesting that the injection of the virus and the premature cell deaths of some Cajal-Retzius cells doesn't really affect the natural physiological reduction of the Cajal-Retzius cells that we have in the brain. So now we have a good transgenic line to live with the Cajal-Retzius cells, and we have a model to ablate the Cajal-Retzius cells from the postnatal hippocampus specifically. And in somehow we can say that we are making the hippocampus a little bit more like the neocortex, right? Where the cells are dying soon after birth. So of course, the question now is, what is the effect of such an ablation? So for this, we kind of got inspired a bit by the literature that was on neocortical Cajal-Retzius cells, which especially the group from Garel and Pierani in Paris, they have shown that the changes in the density of Cajal-Retzius cell can affect the spine density of pyramidal cells. So Ingrid Glarum first joined my group as a master's student, and she's now doing her PhD, really took over this enormous task of imaging terminal dendrites in SLM, radiatum, and oriens from CA1, and counting and classifying spines in mushroom, thin, stubby, and filopodia. And for this talk, we will concentrate only on mushroom and thin spines. And for those of you that are not very familiar with the different types of spines, what the literature suggests is that mushroom spines are slightly more mature spine, and they're being connected more with memory, while thin spines are more slightly less mature spine, and they seem to be more important for learning. So for example, it has been said that the more the amount of thin spine is correlated with the capacity of plasticity of the network. So we started to look at at the spine in stratum lacunoso molecolare, because this is where the Cajal-Retzius cells are. So we would have bet that, you know, this is where we're going to see something. And of course, we didn't see anything in this layer, which was slightly disappointing. But then we started to look at the other layers of the hippocampus, and here very interesting things are starting to appear. So if we look in the radiatum and the oriens, we can see that the total number of spine decreases. And this decrease in the number of spine is driven by the thin spine, which are really decreasing quite a lot in number. And then, you know, interestingly, we also have a change in the specific type of spines that we have, because in the oriens, and this is the only group the only layer where we've seen this, we have a slight but very significant increase in the number of mushroom spines. So this was really exciting, because we said, okay, this is really a global effect. This is not something very specific of one layer. But then we also thought a bit like, why do we see these? We see no changes in SLM. And one of the things that I didn't tell you in this case is that the analysis that we have done was actually spanning the 4th and 5th postnatal week. So we collected this data between P22 and P35. And we knew, it's known from the literature, that the density of spines changes in postnatal development. And so we thought to correlate the density of the spines with the postnatal age. And here things really started to be interesting. Here is the analysis in stratum lacunoso molecolare. And if we concentrate first on the blue lines, which are the PD-1 secret negative animals, so the control animals, we can see that in postnatal development, there is an increase in the spines, particularly in the thin spine, right? But then if we look at what happens when we remove the CAR2 cells, and again, the thin spines are the most interesting here, we see the completely opposite trend. And not only there is an opposite trend, and so the CAR2 cell decrease, but the spine decrease. But what is also very interesting is that at earlier stages analyzed at P22, we actually have more thin spines, and then they just drastically drop in the span of 10 days. And then when we did the same analysis with the radiatum and the orans, they're very similar here, so we can just go through together. We saw again that the thin spines are the ones that are increasing the most in density during postnatal age, and these, again, in the blue we have the control, but then we have pretty much a flat line in the Cre-positive animals, so there is no positive correlation here between the postnatal stage and the density of spine. So this was very exciting for us because we were able to show that we have changes in the spine, in the subtype of spine, and this is not something that is really localized to SLM, but it's something more global, suggesting that we're really, the presence of the CAR2 cell can significantly affect the maturation of possibly the whole hippocampus. So with this data in mind, in our hands, we wanted to understand a little bit better maybe the molecular underpinning supporting these changes. And so in collaboration with the lab of Barbara Van Loom and Paul Satrum at the Department of Cellular Molecular Medicine here at NTNU, we decided to perform a bulk RNA seq from the dorsal hippocampus from injected mice. So we just isolated the hippocampus, took the dorsal part where we saw the fluorescence of the virus, and then perform bulk sequencing. Keegan Dunville, a postdoc in my lab, a really talented postdoc in my lab, performed the analysis of this data. And when we look at a heat map that correlates the expression of genes in the different groups that we have, we can see that the first main divider of the groups is the age, right? So we have the P30 animals on one side and the P15 animals on the right side. We did these 2 time points because we really wanted to see the changes in the potential changes in developmental trajectory. But then when we start to look a little bit at the genotype, right, so the presence or not of CAR2 cell, you can start to see the changes in the gene expression. So because we saw these, we didn't want to remove the developmental component from these analyses, we decided to concentrate on what we call the delta Cre. So the delta Cre negative is the comparison between the 2 time points in Cre negative animals, and the delta Cre positive is what we call is the comparison between P30 and P15 in the Cre-positive animals. So we perform synaptic gene ontology analysis with this single software, and here you can really see in this pie chart different components of the synapses, right? So we have the postsynaptic mainly on the right side and the presynaptic on the left side, and the different components of this part. And when we start to concentrate on the delta Cre negative, so our control, we can see that we have quite a few changes in the gene count. So we have a lot of genes that are related to synapses that are changing their expression between the P30 and the P15 group. And when we look at the enrichment analysis, we can see that these changes are actually significant. When we compare these pie charts with the Cre-positive group, so where we have ablated the CAR2 cell, we actually see that we have a decrease in the gene count present. So we have less genes that change in expression, and these changes are not significant. So really, this suggests that a lot of genes that are related to synapses that normally would change in expression during postnatal development fail to do so when we remove the CAR2 cells. At this point, we wanted to look a little bit more in what these genes are. Can we pinpoint on something, more substantial? And when we did a volcano plot of the delta Cre positive, we started to pick up some very interesting gene. So here on the volcano plot, you see on the right in red all the genes that are upregulated, and on the left you have all the genes that are downregulated. And you can see here, it's a bit small, but hopefully with the arrow it helps. We have this gene called Lactofeeling 2. So this gene normally is downregulated in development. We also observe these in our sample, but it's 8 times more downregulated in the Cre-positive group. So what is Lactofeeling too? So Lactofeeling has been studied mainly by the Leach and Luo lab and Tom Sudoff lab at Stanford, and they were able to show that this gene is very important for the correct connectivity of the entorhinal to hippocampal fibers. And so we wanted to see if these change that we seen and we saw in Lactofeeling was specific for our region. And so we perform a microdissection of the SLM, stratum lacunoso moleculare, from CA1 at P15 and P30, and perform RT-PCR to quantify the changes in the expression of Lactofeeling. And what we saw was really interesting. We can see, so if we focus first on the Cre negative, we can see that the protein normally goes up in the expression in postnatal development. But then in the Cre positive, actually, we have a downregulation in these expressions. So if we compare the 2 time points, we have more Lactofeeling at P15, and then we have less Lactofeeling at P30. And if you remember what I showed you about the thin spine, this correlates really well with what we observe. We have more thin spines in early stages of development, of postnatal development, but then we have less spines at the end. So really, it seems that these changes in the expression of Lactofeeling could support or really be correlated with the changes in the spines that we see. We also perform immunohistochemistry for Lactofeeling to see if at the protein level we could see a difference. And then you can see here in the control animals, you have a really beautiful staining, very localized in SLM with this shade from medial to lateral. And in the Cre positive animal, the expression is severely reduced, so it's almost completely gone, suggesting that also the protein level Lactofeeling is gone. And then lastly, we wanted to see if we could correlate these changes that we have seen in the these layer-specific changes that we observe in spines at the in the synaptic physiology. So we patched C1 pyramidal neurons while stimulating glutamatergic fiber, putative entorhinal cortex fiber from stratum lacunoso moleculare, and we recorded the AMPA and NMDA current. Here in gray, you have the AMPA current, which was reported at -70 millivolts, and then we depolarized the cell to +40 millivolts in the presence of AMPA receptor blockers to record the NMDA current. And then what we did was very simple. We just calculated the peak of the current, and then we did the AMPA and NMDA ratio. And you can see here that we actually have an increase in the AMPA and NMDA ratio. And this, of course, we don't know which one of the two sides is actually changed in this case, because these experiments were not really built and cannot really answer the question. But we know from the literature that changes in the AMPA and NMDA ratio have been correlated with changes in the integration properties of dendrite, and potentially also changes in plasticity because of changes in calcium entries in the dendrites. And the other very speculative but interesting thing that I wanted to point out is that from the literature, it has been suggested that we do know that spines that are more mature have more AMPA receptors than NMDA. So in theory, mushroom spine should have more AMPA receptor and thin spine should have less. So the fact that we have a higher AMPA and NMDA ratio could nicely correlate with what we observe again in the spines, where we have less mature spines, therefore suggesting that the ones that are left over are more mature. So therefore, the higher AMPA and NMDA ratio. And with this, I hope I convince you that the ablation of carotid really have a big effect on the hippocampus circuit. We are able to observe layer-specific changes in the density and type of spines. We can see changes in gene networks related to synaptic plasticity, and here we were able to identify Lactofeeling, which has been shown to be really important for the layer 3 entorhinal cortex 2 CA1 SLM connectivity. And we also show that these changes at the spine level are correlated with alteration in synaptic physiology of CA1 pyramidal cell. So altogether, the data suggests that the Cajal-Retzius cells really have a crucial role in the postnatal hippocampus for the development of the entorhinal hippocampus circuit. So hopefully I have still one last minute. I just want to throw at you some of the future direction that we're working on in the lab. So one of the things that we want to understand is what are behavioral changes that we can observe. So for example, do we see, are the spine's alteration correlated with deficits in learning and memory? And so therefore, Ingvild in my lab now, she's performing behavioral tests for hippocampus dependent learning. We also want to record from spatially modulated cells. We know that there are place cells in the hippocampus and pre-cells in the medial entorhinal cortex. So we will implement neuropixels recording for these experiments. And then we really want to understand a little bit better what is the mechanism supporting these changes, and if these changes are really more global in the whole hippocampal formation. So we're thinking to implement spatial transcriptomic or single cell RNA seq, or a combination of both, to try to understand what are the different passages and the mechanism guiding what we observe so far. And with this, I will just conclude and thank people in my lab, especially Ingvild and Keegan, who has worked really a lot on this project. My collaborator at the Cajal Institute and at the ICOM department here at NTNU, and all the funding resources. And of course, thank you so much for the attention. And I'll be happy to take any question now.